Let's go back to, let's start by reviewing, okay? I'll do glycolysis and then I want you guys to do the other steps. So in glycolysis, we started with glucose. And then we ended with 2 pyruvate molecules. And the potential energy that we got out of that, or molecules with potential energy, were NADH and ATP, and we got 2 of each of those, right? Okay, so what's the next step? Pyruvate oxidation. What happens in that? What are the energy molecules you get out of that? And so on and so forth. I'll give you a few minutes to see what you remember and fill in the blanks from that. NADH, right. And how many NADH do we have? 2. We have 1, but we run it through twice because we have 2 pyruvates, so we have 2. Yeah.
Ask them a question.
Yes.
How do we know that those have potential energy?
Okay, so we started with the NAD+, and then we added a bond, and we added that hydrogen onto it. So that bond is the potential energy. Yep. Okay. Okay, so what's the 4-carbon molecule that we add to acetyl-CoA? And then what do we get? Citrate, or citric acid. And then there was one other one we had to know. Okay. All right, and then what did we get out of this? So how many ATPs for one cycle? 1 times 2, because we run it through twice. So we get 2 ATPs. How many NADH?
6, right?
Yep, 3. And we run it through twice, so it's 6. And how many FADH2s? 1, we run it through twice, 2. Oops. Okay, so this is all review. You guys have this memorized already. Old stuff. You're ready for some new, invigorating material, right?
Yeah.
Pumped. Okay. So what we have, we've got potential energy in this hydrogen, these hydrogens. Oops, I'd have ATP written twice. Okay, so we've got some potential energy here, here, and here. It's also on the ATP, but that's already ready. We can use ATP for energy, so that's ready to go. We got 4 total ATPs out of these processes combined, but the bulk of the energy, or the bulk of the ATP, is produced in these final steps. Okay, so the final step is, okay, so this slide, so each of these NADHs can be converted into how many ATPs? 3. All right, and this FADH2, how many ATPs? 2. Oh, sorry. NADH3, FADH2, right? So how many total ATP do you get out of this whole process? After you run these through oxidative phosphorylation, how many ATP do you get? Add it up. Theoretically. Do the math.
36, right?
Okay, 36, 38, anyone else? Everyone else just waiting for the answer? Okay, 38 is correct, right? 2 times 3 is 6, plus 2 is 8. 2 times 3 is 6, so 14, 16, 6 times 3 is 18, plus 2 times 4, 38. I didn't actually do those last two steps, but it equals 38. Okay? That is the maximum theoretical yield. In actuality, not all those protons go through ATP synthase. Some of them find a way through some other way. And so you actually only get 30 to 32 ATP out of one molecule of glucose. Okay? Oxidative phosphorylation. Okay? And it's just, and that describes what you're doing. Oxidative, you're using oxygen. We'll describe what oxygen is used for. And you're phosphorylating ADPs. You're adding a phosphorus onto ADP in these steps to get ATP. Yeah, no. Okay. So, if you remember, we have our NADHs. And actually what we're going to do is we're going to oxidize them to NAD plus again. And that releases an electron. And what you have here is a series of proteins and enzymes that can take that electron and pass it from one to the next in a series of redox reactions. Okay, redox meaning something is reduced and the other thing is oxidized. Okay, as you do that, it passes, it makes energy available for protons. Okay, H plus ions, hydrogen ions, which are just a proton by itself. And as those electrons are being passed through these redox reactions, that provides energy to move hydrogen ions from the mitochondrial matrix to the intermembrane space. Okay, so NADH, you can do this. You can also do this with FADH2. But FADH2 occurs at a later stage in the process, so you can't get as many protons across with FADH2 as you can with NADH. So, is this an active or passive process? Active, right. So we aren't using ATP, but instead we're using energy provided by the passing of this electron through these enzymes. Yes?
So what causes the hydrogen to, the electron to split off from the energy?
An enzyme. It's this protein embedded within the inner mitochondrial membrane has enzymes which, puts it in the arrangement to where it can split off, But at the end, you still have this electron, and an electron by itself is pretty reactive. And so what we use then to cause it to not be reactive anymore is oxygen, and that's why it's called oxidative phosphorylation, because we use oxygen as the electron acceptor at the very end. And when it accepts those electrons, it makes water. And so that's the final part of our equation. We already had carbon dioxide produced in the previous steps and water at the very end when the electron is no longer needed and is attached to the oxygen. Okay, this process, which I just described, is only half of it, right? So I'm going to make this smaller. This half is the electron transport chain. Okay? What we have then is we have now all of these protons which are built up in the intermembrane space, and they want to flow from their greater concentration to lower concentration. So we've built, again, more potential energy. And what we do then is using this enzyme, which I just drew funnier than the other ones. It doesn't really look like that, but something like that. It's called ATP synthase. And it uses the energy from the protons passing through from greater to lower concentration to make ADP into ATP. Right? And that's the whole purpose of this thing. This is the very end. Making ATP. And so this part then is called chemiosmosis. So the whole part, both together, are oxidative phosphorylation. The part where you have the electron being passed through the enzymes, through the proteins embedded in the intermitochondrial membrane, is the electron transport chain. At the very end, when you're passing protons from greater to lower concentration through ATP synthase, making ATP, that is chemiosmosis. Yes?
So is this ATP synthase turning the hydrogen into phosphate to add to the ATP?
No, the phosphate is there. I just didn't draw it in there. But all it is the hydrogen passes through and it gives the energy to add that bond between a phosphate and ADP. Yeah.
What do we do with the hydrogen afterwards?
It can be reused, be pumped back up.
Cool.
Yeah. So from NADH, because it's used earlier in the process, it can make 3 ATPs for every NADH. FADH2, because it is occurring later, it can only make 2 ATPs.
So did I get that was a ratio of 1?
I'll go back. So this is the end. Now you can start from a glucose, go through all those steps, and get ATP at the end, right? But some organisms, okay, so we'll move on. So that cannot do that whole process. They don't have mitochondria, or they don't have the membranes to allow it to happen. So instead, they just use glycolysis for energy, right? So that's what I drew here on the left. And glycolysis gives you ATP. It doesn't give you very much, but it gives you quick ATP that you can use right away. In order for glycolysis to continue, though, you need NAD+. So if you are continually doing glycolysis, eventually you will run out of NAD+, and so you need more NAD+. You'll have lots of NADH, but that won't do you any good. So, fermentation then takes pyruvate and in a few steps converts it into ethanol or lactic acid. It's one or the other, and there's actually different types of fermentation as well, but these are the two main ones. So, Humans, you and I, we use lactic acid fermentation. Okay? But the purpose of this is in the process of converting pyruvate into lactic acid or ethanol, you oxidize NADH back into NAD+. So now you have NAD+ available for glycolysis to continue. You don't get any ATP out of the fermentation side, but you regenerate NAD+ so that you can keep on doing glycolysis. Okay, pyruvate, however, is a 3-carbon sugar, and ethanol and lactic acid are 2 carbons. So what do you think happens? Yeah, you get a carbon dioxide from fermentation. And so yeast, which does fermentation, if you've baked with yeast before, it creates bubbles. Those bubbles are filled with carbon dioxide.
So I've been told that like our muscles produce lactic acid, and that's what.
You guys know this, right? This is what you guys just proved to me with your stellar drawings out on the chalk. You know this beginning to end, but there are other ways to get energy, right? You don't just eat sugar all the time, right? Maybe you do. I don't know. I lived off marshmallow mateys my freshman year of college, but there are other ways, and so let's start with fats, right? What are fats made out of? Okay. Triglycerides, what are they made out of? Glycerol and fatty acids, right? So, how many carbons does glycerol have? 3, right? Yeah, so where would it fit nicely in here? Yeah, so actually, glycerol can be converted into an intermediate of glycolysis, and then can fit right in to the process
What happens to the
before. Well, the fatty acid, great question, goes through a process called beta oxidation, which actually takes off, fatty acids have a long string of carbons, right?
Yeah.
Beta oxidation takes off 2 carbons at a time and then converts them into acetyl-CoA. And so and that's very efficient because with glycolysis, right, or with glucose, you start with 6, but you lose one of them to carbon dioxide in pyruvate oxidation. Or here, you can just take the 2 carbons, run them through the citric acid cycle, and you get more energy per carbon with fats, with fatty acids, than you do with glucose. Okay? All right, so that's what you can do with the fats. But on the other side, you have proteins. Right, you got your protein shake. And what are proteins made out of? Amino acids, right? And these have various structures, but all of them have an amino group on them. An amino group is not good for using breaking down into energy. So we have to get rid of that amino group, which is NH3, okay, in a process called eternal deamination. It's just deamination. It's just my biology joke. It goes over very well at SVU as to other secular schools. They don't get that stuff, right? Okay, once you take off the NH3 group, now you have a string of carbons that you can use, and depending on what the structure is, it may be an intermediate of the Krebs cycle or acetyl-CoA or
What did you say?
one other thing.
What goes to those?
So whatever's left over on the amino acids. So there's 20 different amino acids. They have all sorts of different structures, so they can be converted into different intermediates. Okay? And that's the whole story. So this is another great figure to know. Okay? You know all the stuff in the middle. We're just adding a few things on the sides This is how we also get energy out of other macromolecules. Yes?